Okay. As I was saying, the inner layer of the cavity, the tissue, always the tissues lining the cavities, we have epithelium, our epithelium, okay? So we always have epithelium lining cavities. Whenever we have cavities in the body, we should have an epithelium lining this cavity. So the inner layer of the uterus is composed from epithelial cells. It is an epithelium and it is called the endometrium. And this endometrium is a glandular epithelium. So it is an epithelium containing glandular cells. Pseudostratified epithelium composed from basal cells and single-layered columnar columnar cells. But as you know that the pseudostratified epithelium is a single-layered epithelium. So it is an epithelium composed from one layer of cells containing basal cells, the short triangular basal cells, and taller cells, which are the columnar epithelial cells. Okay. And we have also in this epithelium glycogen secretory cells. And those secretory cells, they secrete glycogen that helps in nurture in the nutrition of the zygote during the implantation. Okay. So glycogen is used as a nutrition of the zygote during the implantation. And the columnar epithelial cells are ciliated. So this epithelium, this endometrial epithelium is constituted from ciliated cells, ciliated columnar cells and basal cells, and it has also glycogen secretory cells. Now, this is at the level of the uterine body, but at the level of the cervix, we also have a pseudostratified epithelium composed from basal cells and columnar ciliated cells, but we don't have glandular. We don't have glandular cells, so we have mucus secretory cells. Okay, so at the level of the cervical canal, at the level of the cervix, the endometrium is composed from columnar cells, columnar ciliated cells, and basal cells, and mucus secretory cells. So now we don't have cells that secrete glycogen, but we have cells that produce a mucus. Okay, and this mucus have the consistency of this mucus has a role during the fertilization. So when this mucus is thick, it doesn't allow the entry of the sperm cells, and when it is more liquid, it helps, or it permits the entry of the sperm cells into the uterus. And the thickness of this mucus is regulated by the estrogen. We are going to see this in the next chapter, but just to understand why this, why do those cells secrete mucus, why we have mucus at the level of the cervix, and what does it serve for. And also this mucus is important because it protects the uterus from microorganisms and bacterial infections. Okay, so due to the presence of the mucus, they constitute a barrier to the entry of any microorganisms inside of the uterus, and this will help in protecting the zygote or the newborn, the developing fetus inside of the uterus. Okay, so this is regarding the uterus. Now, under the uterus, or in the lowest part, we have the vagina. Okay, so the lowest part of the uterus is the cervix. Then we will have the vagina. The cervix opens in the vagina, And the vagina is a muscular canal. It leads from the uterus to the outside. So it leads from the cervix or from the uterus to the outside of the body. And it is in the vagina that the female receives the sperms from the sperm cells from the male during the coitus. Okay, so the sperm cells are released inside of the female, the vagina, the female's vagina. And then from the vagina, they will move, the sperm cells will move to the cervix, then to the upper parts of the uterus, and then to the fallopian tubes. So this vagina is part of the, and this vagina is part of the birth canal during the birth process. Okay, since the child will, should pass by the vagina during the birth process, and it contains it is an excretory canal for the products of menstruation. So, as we have said earlier, the menstruation are due to the shedding of the endometrium. So during the menstruations, the endometrium will be shed, and we have blood vessels with them that will be degraded. That's why we will have blood that goes out. Okay, and the vagina, so the blood will goes out from the uterus, will reach the vagina, and then the outside of the body. So the vagina is part of this is an excretory canal for the products of menstruation. Its upper part connects to the cervix of the uterus, and so the upper part of the vagina is connected to the cervix. Now, from what is composed the vagina? The vagina is composed from a stratified squamous epithelium. So from the now this is the inner part. So the inner layer of it is composed also from three layers. The inner layer is a stratified squamous epithelium. And this epithelium, as you can see, it is squamous and it is not pseudostratified. So it is a stratified squamous. We don't have columnar cells here. We have squamous cells, and it is a stratified epithelium. And normally, we find the stratified squamous epithelia at the surfaces where we have tensions or pressures. Okay, so it can handle pressure or some movements. Okay, and this is due to because the vaginal epithelium is confronted to the during the coitus. They will have some pressure on the cells and on this tissue, and that's why we have a stratified squamous epithelium in the vagina. And this epithelium stores the glycogen in response to estrogen. So the cells of this epithelium, they are not secretory cells, but they store glycogen. They don't secrete it, okay, but they store the glycogen under the action of estrogen. And this glycogen will be late when the cells will die. They will release the glycogen in the vagina. And as you know, the vagina is open to the outside, and all the surfaces in the body open to the outside, like the nose, the intestines, the mouth, contains bacteria, okay, microflora or microbiota. So there are bacteria in the vagina, and of those bacteria we have the lactobacillus. And those lactobacillus will use this glycogen rejected to the outside when the cells die as a nutritive element. And when they use this glycogen, they will acidify, they will produce acidic products, and this will lead to the acidification of the vagina. Okay, so the presence of the glycogen, or the glycogen, is used by the lactobacillus of the vagina as a nutrient, and due to the use of the glycogen, the lactobacillus can acidify. Yeah, we will have an acidification of the vagina, and this acidic pH of the vagina will protect also the vagina and the upper uterus, the upper parts of the female reproductive system from other microbial infections and from شو بدنا and from yeast infection also. Okay, so the acidity of the vagina is important to protect this vagina and the uterus from other bacterial infections and from yeast infections. Okay. And the second part of the vagina is a thick and elastic muscle wall. So as you can notice, always from the inside, we have a layer of epithelium. Wherever we are, if we are in the fallopian tubes or the uterus or the vagina, always lining the cavity. So the inner part is an epithelium and the middle part is a muscle mass, is a muscular mass. So it's composed from muscles, and the outer layer in the uterus and the fallopian tube is the serosa. Now, in the vagina, this inner layer is composed from squamous epithelial cells. The inner layer. The middle layer is thick muscular mass, and then the outer layer, the most outer layer, is a sheath of connective tissue that consists of blood vessel, lymphatic ducts, and nerve fibers. Okay, so this is the outer layer of the vagina. Now, the female, now this is regarding the inner reproductive system, because if you remember, in the beginning, we have said that we have the internal genitalia and the external genitalia in the female reproductive system. Now we were talking about the internal genitalia. We will see now the external genitalia. The external genitalia from the outside to the inside are composed from a mons pubis, which is a cushion of fatty tissue covered by skin and pubic hair, and it lies over the pubic symphysis. Okay, in fact, this is the mons pubis. This part is the mons pubis. Then we have the labia majora and the labia minora. Okay, so those are fleshy folds. And then we have here the opening of the urethra, and this is the opening of the vagina. Okay, so this is the opening of the vagina. And this part called the vestibule, where we have between the minora lips and where we have the opening of the urethra and the vagina, is called the vestibule. And here we have the clitoris. And we have also some glands called the vestibular glands. They are two pairs of glands. One pair is the Skene's gland, and is called the Skene's gland, localized below to either side of the urethral orifice, and they secrete a small amount of fluid and is similar to the prostate gland in males. Okay. In fact, those glands serve for lubrication. Okay, so it's a lubricating fluid to facilitate the penetration of the penis during coitus. And the other gland is the Bartholin gland, localized at each side of the vaginal opening. Okay, they secrete also a mucus and are homologous to bulbourethral glands of the male. So we will have the opening of the Bartholin's gland, which is here, localized behind the vagina. Okay, and the openings of the Skene's glands will be next adjacent to the urethral opening. Now we will move to talk about the ovarian functions and to talk about the oogenesis or the folliculogenesis now. So what are the functions of the ovary? In the ovary occurs the oogenesis, as you remember, which is the production of gametes during the fetal period. So in the ovaries, we have the production of gametes, and this production of gametes occurs during the fetal period. Now, those gametes are produced during the fetal period, but their maturation we have also their maturation in the ovaries, and this maturation will take place or will happen only after puberty. Okay, and not all of the oocyte will get to mature at the same time. No, we have menstrual cycles, and at each cycle, only few of those oocytes will start, or follicular oocytes will start to mature, but only one of those of the follicles will continue its maturation. We have also the ovulation. So in the ovaries, we have the ovulation, which is the expulsion of the mature oocyte. Also, the ovaries are the site of the steroidogenesis, which is the production of estrogen and progesterone. So in the ovaries, we will have the production of estrogen and progesterone. And also the ovaries produce other hormones like the inhibin and the activin, okay, and they produce them and they secrete them. So it is the place where the steroidogenesis occurs, and most of the estrogen and progesterones in the female are synthesized, and also other hormones like the peptide hormones, the inhibin and activin, are produced and secreted. 1 second. Now, as we have seen in the first chapter, the oogenesis starts in the ovaries and is achieved in the fallopian tubes upon fertilization. So the oogenesis is only achieved, or the maturation, the full maturation of the oocyte at the end of the meiosis will be only achieved after the fertilization, if it happens. Okay. Now, before birth, a female has starts by having gonia, or oogonia, that develop from the primordial germ cells. So we have primordial germ cells, then they will divide mitotically to give oogonia, and those oogonia also will divide by mitosis. Between the 2nd and the 7th month, they will divide extensively, and we will have by the 7th month almost 7 million of oogonia. Okay, now at the 7th month, most of those oogonia, or most of them, will degenerate, and the remaining one will enter the meiosis and will become primary oocytes. Okay, so whenever those oogonia start the meiosis, we will call them primary oocytes. However, this meiosis will be arrested at the diprotein stage of the meiosis 1, and and all of this will occur before birth. All the oogonia will become primary oocytes, and all of the primary oocytes will be arrested at the diprotein of meiosis 1. So and they will remain arrested in the diprotein of meiosis 1 until puberty. Okay, so when a female is born, it will get born with a pool or a certain number of primary oocytes. So it will get born with a pool of primary oocytes, but not oogonia. So it's not like in the male. A male has spermatogonia, which are stem cells that can always generate other spermatocytes and more mature cells. However, the females will be born with primary oocytes, which are already which are cells engaged in their maturation. So they are not the stem cells, the very stem cells of the gametogenesis. And those primary oocytes are blocked in the meiosis 1. And so we don't have, after birth, we will not have any more generation of primary oocytes, okay? Now, and those primary oocytes are diploid cells, each containing a set of 46 chromosomes with 2 chromatids. Now, after birth, an ovary will contain 2 to 4 million of primary oocytes. But also, many of those oocytes will degenerate during the time before puberty and after puberty. A lot of them will degenerate, and only a few of those 2 to 4 millions will be ovulated in a woman's lifetime. So in a woman's lifetime, only almost 400 primary oocytes will be ovulated, and the others will degenerate. Okay, so at each menstrual cycle, she will have one primary oocyte, one oocyte, let's say, because it will be secondary during the ovulation. So at each menstrual cycle, she will have one oocyte ovulated. And from the puberty time to the menopause, she will have almost 400 of those oocytes that will be ovulated. All of the others will degenerate at some point during their development by atresia, so they will die. And 90% of ovarian follicle present at birth will undergo atresia. So 90% of the ovarian follicles will undergo atresia. The ovarian follicles are the oocytes, the primary oocytes, when they are surrounded by follicular cells, as you are going to see. So the oocytes will not be like this alone. They will always be surrounded by other cells called the follicular cells or the granulosa cells. Okay, so 90%, and this the oocytes and the follicular cells, they make together what we call an ovarian follicle. So 90% of the ovarian follicles present at birth will undergo atresia at some point during the life of a female. And all of the follicles can undergo atresia, because also the follicles will have different stages of maturation. We have the pre-enteral follicles and the enteral follicles. So all of them, they can undergo atresia, and not only the enteral follicles, but the pre-enteral also. So this atresia can occur at any stage of development. By the time a woman reaches approximately 50 years of age, only few ovarian follicle will remain. Okay, and that's why the female will reach the menopause, which is the age at which they will not have any more follicular ovarian follicles to be ovulated. Okay, all of them will be degenerated. So this pool of primary oocytes they had at birth, by the age of menopause, either they will be ovulated or they will die. So by the menopause, they won't have any more of ovulation. And the oocyte will remain arrested at diprotein of prophase 1 until puberty. Okay, so as we have said, at birth they have primary oocyte. Those primary oocytes are arrested at the meiosis 1, and they will remain arrested at the meiosis 1 until puberty. At puberty, not all of them also will resume the meiosis. At each menstrual cycle, only the selected one selected ovarian follicle, one selected oocyte will resume the meiosis, and the others will not be able to resume the meiosis. So only the ovarian follicle or the oocyte destined for the ovulation will resume the meiosis. Now, after puberty, only primary oocyte destined for ovulation will complete the first meiotic division. Okay, so the result, this is after puberty. The result of this first meiotic division is one secondary oocyte, which is a haploid cell, and one polar body, another smaller cell. So we have an unequal division that happens at the level of the primary oocyte, which give one big cell and one small cell. That's because the spindle is not situated in the middle of the cell, and then the constriction will not be in the middle. So we will have one big cell and one smaller cell called the polar body. This big cell is the secondary oocyte, and this secondary oocyte will start the meiosis 2, but then will be arrested at the metaphase of this meiosis 2. And it's only, it will be ovulated as such, so as a secondary oocyte arrested at metaphase of meiosis 2. If we have a fertilization, so if the secondary oocyte is fertilized by a sperm cell, the meiosis will resume again. And we will see the process, we will see the mechanism how and why the meiosis will resume again after fertilization. And then this second meiosis can be achieved, and we will have a oocyte and a second polar body as a consequence of this meiosis 2. Okay, the maturation of an oocyte will only be achieved if there is a fertilization. If there is no fertilization, this secondary oocyte will be ovulated, but if there is no fertilization, it will degenerate as a secondary oocyte blocked in meiosis 2. So it will remain blocked in the meiosis 2, and the maturation of this oocyte will not be accomplished. Okay. Now, the follicle is the fundamental reproductive, or the ovarian follicle, what we call the ovarian follicle, is the fundamental reproductive element of the ovary. After birth, a female has primary oocyte. Primary oocytes reside inside primordial follicles. We will see this. And the primordial follicles, now, as I was saying, this is inside of the ovary. We have the oocytes, okay? And those oocytes, as we have said, they are surrounded by follicular cells. Now, the most immature, they are surrounded by follicular cells to form an ovarian follicle. So this is an ovarian follicle where we have an oocyte, and this is of the oocyte, and those are the follicular cells. This is also an oocyte, this big one, and those are follicular cells. And both of them, the oocyte and the follicular cells, they form an ovarian follicle. So this is an ovarian follicle. And this inside is the nucleus of the oocyte. And here also we have an oocyte and granulosa cells or follicular cells forming together what we call an ovarian follicle. So the ovarian follicle or the follicle is the fundamental reproductive element of the ovary. The most immature follicle is the primordial follicle. What we call the primordial follicle is the oocyte surrounded by flattened follicular cells. So when those follicular cells are flattened, we call them, in fact, we call this follicle a primordial follicle, and this is the most immature follicle. Then at later stages of follicular development, we will see that those flattened cells will become cuboidal, okay, and we will have one layer of cuboidal cells, then more than one layer, and other differences. So the most immature one is a primordial follicles, and those primordial follicles have flattened follicular cells called granulosa cells. And in fact, as you remember, when we talked in the chapter 3 of the gonadogenesis, we said that the follicular cells are of mesenchymal origin, okay? So when we say flattened mesenchymal cells, we mean the follicular cells, okay, that we call them granulosa cells. The earliest signs of follicular growth are when we have follicular growth, we will see a change in the shape of the surrounding granulosa cells from flattened to cuboidal, as I was explaining here. So those flattened follicular cells will become cuboidal, okay? And we will have multiplication of the number of layers of those granulosa cells. So we will have more than one layer. So this is also another sign of the maturation of a follicle. We will have also an increase in the size of the oocyte. Look at the oocyte here in the primordial follicle and the oocyte here in the primary follicle, okay? So we have an increase in the size of the oocyte. This is another sign of the maturation or follicular growth. Those are the signs of the follicular growth. So we have an increase in the size of the oocyte, a change in the shape of the surrounding granulosa cells from flattened to cuboidal, an increase in the granulosa cell number, and the appearance of what we call the Zona Pellucida. So also during the follicular growth, at certain moment, we will have the appearance of a Zona Pellucida. And this is Zona Pellucida is a region surrounding the, in fact, it's a matrix, an extracellular matrix surrounding the oocyte. This extracellular matrix is secreted by the oocyte, and it is an acellular matrix, so it doesn't contain cells. And it is rich in glycoproteins. Okay, it is composed from glycoproteins, and this layer of this Zona Pellucida will separate the oocyte from the granulosa cells, and it will become a little bit more important during more mature follicles. In this follicle, this is Enteral Follicle. We have here, look at all of this mass inside. This is the oocyte, and those are the granulosa cells, and this white zone surrounding the oocyte is the Zona Pellucida. So it will become a little bit more important during the development of the follicle. But at the beginning, in the primordial and primary follicle, it wasn't present. So we don't have Zona Pellucida in the earlier stages of follicular development. Then it will appear, it will be secreted later in the secondary follicle. So the appearance of the Zona Pellucida around the oocyte, is one of the signs of the follicular growth also. Is it clear till now?
Yes, doctor.
Okay. Here we have the different stages of follicular growth. Okay. In fact, we will explain the different stages. What is the difference between them? Now, we have said that at a certain moment, this oocyte will secrete the Zona Pellucida that will separate it from the granulosa cells, okay? And as you know, here we have an ovarian follicle inside of the ovary. We have a connective tissue surrounding the follicles. If you remember, we have said last time that we have ovarian follicles here, and between them we have connective tissue, okay? So all of this space surrounding the ovarian follicles is a connective tissue, containing blood vessels also. So, but the blood vessels do not reach inside of the follicle, so they don't enter inside of the ovarian follicles, and they don't reach the granulosa cells, and they don't reach the oocyte. Okay, so somehow this oocyte and those granulosa cells have to exchange to receive nutrients and to exchange nutrients and factors to keep on surviving. Okay, so how do they do if they are not connected to the outside? Uh, how do they keep on surviving? In fact, we have blood vessels that reach this outer region outside of the ovarian follicle. We will have the nutrients that will diffuse inside of the granulosa cells. Okay, and the granulosa cells are themselves connected. They are connected to each others by gap junctions. And also between the granulosa cells and the oocytes, we have some connections between the inner layer of the granulosa cells and the oocyte. Despite of the presence of the Zona Pellucida, they can be connected. So the cells in the inner layer of the granulosa cells and the oocytes are connected to each other despite the presence of the Zona Pellucida. How do they make this? How can they be connected? In fact, the granulosa cells emit cytoplasmic extensions. They have cytoplasmic extensions. And the oocyte, the surface of the oocyte has microvilli. And so they are connected by gap junctions at the level of those microvilli and cytoplasmic extensions of the granulosa cells. So we always have cytoplasmic extensions of the inner layer of the granulosa cells. Those cytoplasmic extensions, they will extend through the Zona Pellucida. This is the Zona Pellucida to reach the oocyte, where they are connected by gap junctions to the microvilli of the oocytes. Okay, so we have gap junctions that allow the exchange of nutrients and of factors between the granulosa cells and the oocytes. And also due to those connections, as we are going to see, the granulosa cells will control the meiosis arrest in the oocyte. Okay, so the granulosa cells, they secrete factors also, and due to those secretions and to those connections through the gap junctions, those granulosa cells will maintain the arrest, the arrest in meiosis 1 of the oocyte. So we have gap junctions that form between adjacent granulosa cells. Also and those gap junctions will allow the exchange of nutrients and factors between the granulosa cells and will allow the intercellular communication, will form some intercellular communication network. And also, as we have said, the granulosa is completely avascular, so we don't have blood vessels that reach the granulosa, neither the oocyte. So all of the ovarian follicle is not vascularized. Okay, the granulosa is not vascularized, and the oocyte is not vascularized. But we have gap junctions between the granulosa cells and between the granulosa, the inner layer of the granulosa cells and the oocyte, which will allow the exchange of nutrients. And also some factors produced by the granulosa cells are passed to the oocyte and act on them to maintain their meiotic arrest until the time of ovulation. Because this arrest of the meiosis 1 will remain until just before the ovulation, and it's only before the ovulation we will have a peak of the LH. Under this action of LH, that the meiosis will resume and the meiosis 1 will resume, and we will have the primary oocyte that will become will achieve its meiosis to become a secondary oocyte. So how this meiotic arrest is controlled? How is this meiotic arrest controlled? In fact, this is an oocyte, and those are the granulosa cells surrounding it. And in blue, we have the Zona Pellucida. The oocyte produce cyclic AMP. And as you remember, the cyclic AMP is a second messenger that will induce a cascade of messengers underneath it. Okay, here in the oocyte, the cyclic AMP will activate the PKA, the protein kinase A, the PKA, okay, which inactivate a factor called the maturation promoting factor. This factor is responsible for the continuity of the meiosis. Okay, so if this factor is inactivated, if this MPF is inactivated, the meiosis will be arrested. So now, due to the presence of high cyclic AMP levels in the oocyte, we will have activation of the protein kinase A, which will induce an inactivation of the maturation promoting factor. So the PKA under the action of cyclic AMP will inactivate the MPF. And due to this MPF inactivation, we will have a meiotic arrest. And those high levels of cyclic AMP are from one side due to the production of cyclic AMP inside of the oocyte, and from another side, some of the cyclic AMP are produced in the granulosa cells, and they migrate through those gap junctions here at the level of the Zona Pellucida. They will migrate to the inside of the oocyte. So when we have generally in the primary oocyte, when they are arrested in the meiosis 1, we have high levels of cyclic AMP that will induce an inactivation of the MPF and consequently arrest of the meiosis at the diprotein stage of the meiosis 1. Now, before, and this, in fact, those high levels of cyclic AMP are controlled by a factor called the OMI, the oocyte maturation inhibitor. Okay, this OMI is produced by the granulosa cells. And the OMI, from one side, it increases the levels of cyclic JMP in the granulosa cells. And those cyclic JMP levels will diffuse from the granulosa to the oocyte, and they will inhibit, when we have cyclic GMP, this will inhibit the hydrolysis of the cyclic AMP, and this will lead to the maintenance of high cyclic AMP levels, and consequently to the meiotic arrest. So, why do we have high levels of cyclic AMP? Because in the granulosa cells, we have the OMI factor, which will induce the high production levels of cyclic GMP. And once the cyclic GMP is produced in the granulosa cells, it will pass through the gap junctions between the granulosa and the oocyte. Okay, they will diffuse to the oocyte, and the presence of those cyclic GMP, of this cyclic GMP, will inhibit the hydrolysis of the cyclic AMP. So whenever we have high levels of cyclic GMP, this will inhibit the hydrolysis and the degeneration of the the degradation of the cyclic AMP, and thus we will keep on having high levels of cyclic AMP. So as long as we have OMI, high levels of OMI in the granulosa cells, we will keep on having high levels of cyclic GMP that will diffuse to the oocyte and through the gap junctions, and will inhibit the hydrolysis of cyclic AMP, and as a consequence, we will have the MPF inactivated, and then the meiotic arrest that will the cells that will keep on being under in the meiotic arrest. However, before the ovulation, we will have a peak of LH. So the anterior pituitary gland will produce the gonadotrope, the LH, okay, which will have a surge. We will have a peak before the ovulation of the LH. So under this peak of LH production, under this LH surge, the OMI, we will have a decrease in the OMI levels due to the presence of the LH, high levels of LH. We will have a decrease in the OMI levels that will lead to, from one side, to the decrease of cyclic GMP levels, and from another side to the closure of gap junctions, okay? And this closure of the gap junctions will inhibit, or will not allow anymore the transfer of cyclic GMP from the granulosa cells to the oocyte, and this will lead to the decrease in cyclic AMP levels in the oocyte. And then when we have a decrease in cyclic AMP, the MPF is activated, and the meiosis can be, the first meiotic division can be achieved to give a secondary oocyte. So under the action of the LH, we will have a decrease in the OMI levels in the granulosa cells. Those OMI, this OMI, it controls the levels of the cyclic GMP in the granulosa cells, and also controls the gap junctions. So when this OMI levels are decreased, the gap junctions will be closed, and no more cyclic GMP can pass from the granulosa cells to the oocyte, and this will lead that the cyclic AMP can be hydrolyzed inside of the oocyte, and then we will have reduced cyclic AMP levels, and then MPF activation. And due to the MPF activation, the meiosis resumes. So this is how the meiosis of the meiosis 1 is arrested. And as you can see, this arrest in the meiosis 1 is controlled by the granulosa cells and by the OMI factor in the granulosa cells, and by also the cyclic GMP produced by the granulosa cells. Is it clear till now?
Yes, doctor.
Okay, I will continue. Now we will move to talk about the different stages of follicular growth, okay, and its hormonal regulation. So at birth, a female, as we have said, it has primary oocytes, and only primary oocytes, but also, as we have precised earlier, that those oocytes are surrounded by follicular cells and are present as ovarian follicle in a form of ovarian follicle. So at birth, a female has primordial follicles. All of the follicles are immature and are called the primordial follicles. So at birth, the female has all of the ovarian follicles, all of the follicles in the ovaries are primordial follicles at birth. Then few follicles may resume development sporadically before puberty, after birth and before puberty. Few of those follicles will develop a little bit, but then they will stop their development. So they will never reach the stage where they will become enteral follicles, okay? They will develop a little bit sporadically and incompletely during fetal life. Yes, and in fact, they will not continue their maturation, okay? Now, it's only at puberty that we have a regular recruitment of primordial follicles to become more mature follicles. So it's only after puberty that at each month, in fact, this is not related to the menstrual cycle. We'll explain this in the next session. But after puberty, those primordial follicles will be regularly become more mature, okay, and so at puberty, those primordial follicles will be regularly recruited to become more, and at each menstrual cycle, one of those maturing follicle, yes, and at each menstrual cycle, one of these maturing follicle can achieve the meiosis 1 and can be ovulated, okay? So at puberty, we have regular recruitment of primordial follicles to become more mature into a pool of growing follicles. And we have 4 stages of development for the primordial follicle before ovulation. We have the primary follicle, then the secondary follicle, the tertiary, and the preovulatory follicle. Now, the primary and the secondary follicle are called pre-enteral follicles because they don't have a cavity, and we call them pre-enteral follicles, and they constitute the pre-enteral phase. And the tertiary follicle and the Graafian follicle, they are called enteral follicles. Both of them are called enteral. Now, the tertiary follicle is called the early enteral follicle, and the Graafian follicle is called the pre-ovulatory follicle, which is the late enteral, okay, or pre-ovulatory follicle. And both of them will constitute, or the presence of those follicles will constitute the pre-ovulatory phase, will be present, or they will be present. We will find them in the pre-ovulatory phase. Now, the pre-enteral phase, or phase where we have the development from primordial follicles to primary and secondary follicles, this is the longest phase. So this development from primordial follicles to give primary and then secondary follicles, it is the longest phase. It takes the most of the time. And then the pre-ovulatory phase, where we have the development of the tertiary and the Graafian follicle. This is the shortest phase in the follicular development. And now, what are changes that happens to the primordial follicle during its maturation into a primary follicle? First of all, the oocyte will increase in the diameter. Okay, so the oocyte will increase in size, and its diameter will increase in size to have a diameter of 60 to 120 micrometers. And this size of the oocyte reached in the primary follicle. In fact, it is the final size of the oocyte. So later on, the oocyte will not increase in size anymore. Okay, so we will have an increase in the size of the oocyte during the transition from the primordial to the primary follicle, but later on, and this will be the final stage of increase in the oocyte size, and later on we will not have any increase in the size of the oocyte. So the growth, this growth in the size of the oocyte is accompanied by the synthesis of large ribosomes and messenger RNA that will be used during later stages of oocyte maturation. So during this increase in size, the oocyte will also produce messenger RNAs and ribosomes that will be used later during the maturation of the oocyte and the follicle. And we will have the appearance of a basement membrane surrounding the follicle, and this basement membrane or lamina propria will be separating the ovarian follicle from the surrounding connective tissue. Okay, so during this transition from primordial to a primary follicle, we will have an appearance of basement membrane. This basement membrane was not present in the primordial follicle, but it will appear in the primary follicle. And this basement membrane will separate later. In fact, the connective tissue surrounding the follicles will be organized into what we will call a theca folliculi, but not at this stage. Okay, so this basement membrane will separate the ovarian follicle from the connective tissue and from the what will be later formed the theca folliculi. But now we don't have, at the primary follicle stage, we don't have the theca folliculi. The basement membrane will separate the ovarian follicle from the connective tissue. And also, as we have precised earlier, the follicular cells, they were flattened in the primordial follicle, and they will become cuboidal in the primary follicle. So we will have in the primary follicle cuboidal follicular cells, and they will form one layer around the oocyte. So in the primary follicle, we will have one layer of follicular cells around surrounding the oocyte, and they will be cuboidal cells. Those follicular cells will be cuboidal and not flattened, which is the case of the primordial follicle. Now, then this primary follicle will have some modifications and will mature to become a secondary follicle. Okay, so during this transition from a primary to a secondary follicle, we will have the formation of the Zona Pellucida, as we have explained here also, that we will have the Zona Pellucida that will be formed between the oocyte and the granulosa cells. So this Zona Pellucida appears for the first time in the secondary oocyte. So it will appear during the transition from a primary to a secondary oocyte. It was not present in the primary follicles. It was not present, and it will be formed during the transition from this primary follicle into a secondary follicle. So we will have the formation of the Zona Pellucida, which is an acellular glycoprotein layer secreted by the oocyte. And this glycoprotein layer will condense around the oocyte to form a translucent acellular layer. So this Zona Pellucida is a translucent acellular layer. It separates the oocyte from the granulosa cells. And also one of those modifications that will happen during this transition from a primary to a secondary follicle is the division of the granulosa cells. They will divide and will form, and will become multiple layer thick. So in the okay, in the primary follicle, we had one layer of granulosa cells surrounding the oocyte. In the secondary follicle, we will have more than one layer. So those granulosa cells that were here will divide to form more than one layer of granulosa cells surrounding the oocyte. And also those one of the modifications that will occur is that the connective tissue surrounding the follicle will be organized in sort to form the theca folliculi around the oocyte. So the ovarian stromal cells are the connective tissue cells. So the ovarian stromal cells on the outer basement membrane will condense into spindle-shaped cells called theca folliculi. So the cells of the connective tissue adjacent to the this was a primary follicle, okay, and this is a secondary follicle. Here we have the stroma or the connective tissue, okay. Here we have the stroma, and here we have the basement membranes separating the follicle, the ovarian follicle, from the outside, from the connective tissue. Now the cells of the stroma surrounding or adjacent to this basement membrane will become more fusiform, okay, and will condense around this basement membrane to form what we call the theca, okay, the theca folliculi. So we'll have a modification in the shape of the cells surrounding or adjacent to the basement membrane of an ovarian follicle, and they will condense to form the theca folliculi. So the ovarian stromal cells on the outer basement membrane will condense into spindle-shaped cells called the theca, or spindle-shaped or fusiform means, okay, called the theca folliculi. Now, how is this development or maturation from a primordial to a secondary oocyte controlled? يعني do the LH and FSH hormones have any role in the development of those of this primordial follicle into a primary follicle, and then to the development of this primary follicle into a secondary follicle? In fact, no. The primordial this development from a primordial into a primary, then into a secondary follicles, is independent. So it's not dependent of the pituitary gonadotropins. It's not dependent from the FSH and LH. And in fact, we have some growth factors produced by the granulosa cells that will control this development, but this development is not dependent from the LH and from the FSH. And those cells, the follicular cells of the primordial, primary, and secondary oocyte, they don't have receptors, in fact, of the LH and of the FSH. And since they don't have receptors for the FSH and the LH, then they cannot respond to their action. Even if we have LH and FSH that are produced, those follicles, the primordial, primary, and secondary follicles, they don't have receptors for those pituitary gonadotropins. They don't have receptors for the FSH and LH. So they are not able to respond to their action. And thus their development is completely independent from the action of LH and FSH. However, at certain moment during in the late secondary follicle, so when this follicle, the secondary follicle, is mature and fully developed, it will start to express FSH and LH receptors, okay? But when it is formed, it doesn't have the secondary follicle doesn't have FSH and LH receptors. Okay, and from the moment where this late secondary follicle will start to express LH and FSH receptors, it can start to respond to the action of the gonadotrophins, yani to the action of the FSH and LH. But before, they didn't used to have whole secondary follicles. They didn't used to have FSH and LH, and they don't respond to the action of those gonadotrophins, okay? And normally the LH receptors will be expressed on the theca cells, so on those cells which are adjacent to the basement membrane, but outside of the follicle. And okay, this is in the early stages of the follicular development. And the FSH receptors will be expressed on the granulosa cells, okay? Now, in the later stages of the follicular development, we will see that under the peak of the LH, the granulosa cells will start to express LH receptors, but this comes later on. So what you should know is that the primordial, primary, and secondary receptors, they do not express LH and FSH receptors, and their maturation is not dependent from the action of LH and FSH. It's only secondary follicle. Once the secondary follicles will undergo a major maturation, they will acquire FSH and LH receptors, and the FSH receptors will be expressed on the granulosa cells, and the LH receptors will be expressed on the theca cells. And also, the late secondary follicles will express estrogen receptors on the granulosa cells, besides of the receptors of the FSH. So the granulosa cells in the late secondary follicles will express estrogen and FSH receptors, and the theca cells will express LH receptors. Okay, but the development, the maturation of those follicles is not, the maturation from the primordial to a primary, then to a secondary follicle, is independent from the action of the gonadotrophins. Now, what comes next? So, as you can see, here we had a primary, a secondary follicle, and then we have follicles that are called the tertiary and the Graafian follicle. What happens, so we have some modification that will occur from the transition of secondary to a tertiary follicle. We will have more granulosa cells first, and those granulosa cells will start to produce a fluid. In fact, so we'll start to have some secretions, and those secretions of the granulosa cells are a fluid containing steroids like the estrogens and the progesterone, because as you are going to see, those cells, the granulosa cells, they are the cells that produce the estrogen and progesterone. So their secretions will contain estrogen and progesterone. And we have also in this fluid some FSH and LH, okay? And this fluid secreted by the granulosa cells will constitute droplets. We will have droplets of fluid inside of the follicle, and then those droplets will coalesce together, so they will group together to form a cavity, filled with a fluid, and this cavity is called the antrum or the enteral cavity, okay? And that's why we start talking about enteral follicles now, and before we were talking about pre-enteral follicles, okay? So from now on, starting from this stage where we have the secretion of this fluid from the granulosa cells, we will have cavity forming, and this cavity will be called an antrum. And now, whenever we can identify the presence of this antrum inside a follicle, this follicle will be called an enteral follicle. Now, the enteral follicles, we will have an early enteral follicle that is also called the tertiary follicle, and we have a late enteral follicle that is called the pre-ovulatory or Graafian follicle. Okay, so now this is a tertiary follicle where we can see we have a small cavity. This is also a tertiary follicle, but the cavity is more important and much bigger, and this is directly the tertiary follicle stage before the Graafian follicle, okay, or it's just close to become a Graafian follicle. Now, also, one of the differences that occur during the development from a secondary to a tertiary follicle is that the theca formed during the theca that we have observed surrounding the secondary follicle will differentiate, and so we will have a differentiation of the theca into two different layers, the theca interna and the theca externa. In the theca externa, we will have myofibroblasts. It will be rich in myofibroblasts, so in fibroblast-like cells, and those cells, they can contract, okay? And the contraction of those cells in the theca externa will help later in the ovulation process, okay? Will help to expulse the oocyte outside of the follicle, okay? So the theca externa is composed from myofibroblasts and is vascularized, okay? And we have the theca interna, where the cells here will become more round-shaped, okay? And and they will differentiate into glandular cells. So the theca will differentiate to give the theca externa and more circular cells, which are glandular cells also. And those cells will constitute the theca interna. And this theca interna is highly vascularized, okay? And in fact, those theca interna cells will produce androgens, okay? And those androgens will diffuse inside of the granulosa cells. And inside of the granulosa cells, those androgens will be transformed into estrogens, okay? So most of the androgens produced by the theca interna will be transformed by the granulosa cells into estrogens and progesterones. So in the tertiary follicle, or between the transition from the secondary to a tertiary follicle, we will have the theca folliculi that will develop into two different layers. In the secondary follicle, it was one layer, only one layer. In the tertiary follicle or the early enteral, we will start to see two layers of the theca folliculi. The theca externa, which is a fibrous capsule, and the theca interna, which is the inner layer of the theca. This is the layer directly adjacent to the basement membrane of the ovarian follicle, and it is a glandular layer and highly vascular, okay? And also we will have the membrane of Slavjensky. In fact, the basement membrane surrounding the basement membrane surrounding the follicle will be called the membrane of Slavjensky. Okay, this one here. So here, this basement membrane, what we call the basement membrane earlier, will be called the membrane of Slavjensky. So whenever you hear a membrane of Slavjensky, this means this is the basement membrane surrounding the follicle. Okay, and this membrane of Slavjensky will separate the theca from the ovarian follicle, and it will be directly adjacent to the theca interna. The membrane of Slavjensky is the basement membrane. It separates the theca interna and externa from the granulosa cells. And in the tertiary follicle, the granulosa cells continue to proliferate and they increase in, so we will have more granulosa cells, and which will make that during now we will have follicular growth due to this granulosa cells division. Okay, and so the follicular growth will be now due from one side to the division of the granulosa cells, and from another side to the presence to the volume of the fluid inside of the follicles, and from another side also is and also this increase in the or this growth of the ovarian follicle will be also due to the increase of the size of the cavity. The more we will have fluid produced by the granulosa cells, the more this cavity will become bigger. Okay, so now in those stages of follicular development, the follicular growth will not be due to an increase of the size of the oocyte, but it will be due to an increase in the granulosa cell number, to an increase in the volume of the enteral fluid, and to an increase in the size of this entrum or of this cavity. Okay. From now on, the increase in follicular size will depend on the granulosa proliferation, the increase in the volume of the follicular entrum, and the increase in the volume of the follicular fluid. The oocyte does not increase in size during the enteral phase, but it continues to actively synthesize RNA and to turn over proteins. Okay, so we don't have any more increase in the size of the oocyte. As you have said, the size of the oocyte will reach its maximal size at the primary follicle stage. بعد the primary follicle stage, we will not have an increase in the oocyte size. Now, from the tertiary to the Graafian follicle, we'll talk about the transition from a tertiary to a Graafian follicle. The enteral or the late enteral follicle will have a fully developed follicular entrum. Okay, they will have an oocyte surrounded by different granulosa cells, the corona radiata, the cumulus oophoris, and the mural cells. Look, this is a tertiary follicle, and this is a Graafian follicle, or the pre-ovulatory follicle, or what we call also the late enteral. Okay, so in the Graafian follicle, we will have the biggest part of the follicle will be occupied by the enteral cavity. It will become so big. This is the oocyte inside of the follicle. We can see that the oocyte will be surrounded by granulosa cells here, and here we have granulosa cells also. Now, the granulosa cells surrounding the oocyte will be called here surrounding it, they will be called the corona radiata. They will form what we call the corona radiata. And then those granulosa cells forming a sort of bridge between the corona radiata and the periphery of the follicle, they will be called the cumulus oophoris. Okay. And then we have here this layer of granulosa cells. adjacent to the basement membrane or to the membrane of Slavjensky, they will be called the mural cells, the mural granulosa cells. So the oocyte will be surrounded from the corona radiata cells and by the cumulus ovorus that constitute like a bridge between the corona radiata and the periphery of the follicle. And we have the mural cells, which are the cells adjacent to the basement membrane. But the most important size or part of the follicle will be occupied by the enteral fluid. Okay, by this enteral fluid. This is a tertiary follicle. We can see that we have an increase in the volume of the enteral fluid, in the size of the antrum, which is much bigger. And here we have the follicular cells organized into a corona radiata, a cumulus ovorus, and mural cells. Okay. We have also the membrane of Slavjensky and the theca interna and externa that will be also present. And they are telling you that the theca externa is a fibrous layer made of collagen fibers, myofibroblasts, and blood capillaries. Okay. Now, the late pre-enteral and early enteral follicles will express FSH and LH receptors. As we have explained earlier also, that those FSH and LH receptors, they will start to appear in the late secondary follicle. So the early enteral and the late enteral follicles will express, sorry. Yes. So the late tertiary follicle and the Graafian follicle will have FSH and LH receptors expressed on their cells, and they can respond to the action of the LH and FSH. Now also, as we have explained, that the LH receptors will be expressed on the theca interna cells. Here you can see this is a follicle, ovarian follicle. This is the connective tissue or the stroma of the ovary. And this is the theca. This is the theca interna and externa surrounding the follicle. We can see how we have condensed cells around surrounding the follicle and how they are a little bit different from the other stroma. Okay, so those are this is the theca and divided into the theca interna and the theca externa. Now the theca interna cells, they have receptors for the LH, and under the action of LH, they will produce androgens, okay? And the FSH receptors are only expressed on the granulosa cells, okay? So the granulosa cells, they only the granulosa cells express the FSH receptors. The theca cells, the theca interna cells, don't have receptors for the FSH. Okay, and now how did we have this maturation from a secondary to early enteral and then to a late enteral follicle? Since those cells that I have mentioned now express receptors for the FSH and LH, they can respond then to the action of those two gonadotrophins and it's under the action of the LH and FSH that they are going to mature. But in fact, the studies showed that it is the FSH that is the most responsible for this maturation of the follicles, and so to start the maturation from a late secondary to an early enteral, we need FSH, not LH. Okay, so this first step of maturation from a secondary, late secondary to a tertiary follicle, it is made under the action of the FSH alone. Okay, that's why they are telling you that the FSH alone is sufficient for the early follicular growth. But later on, during the later development from the early enteral to the late enteral, the LH will be also responsible for this follicular growth. Okay, so the FSH alone is sufficient for early follicular growth, but the LH will assist for further follicular growth. The LH will assist for the further development of the follicles from the enteral to the late enteral. If there is no LH, this transition from enteral to the late enteral will not happen. So the FSH alone is sufficient for early follicular growth, but the LH will assist for further follicular growth. And the gonadotropins will pick up the pre-enteral follicles and stimulate the enteral growth. So the gonadotropins will act on the late secondary follicles. That's why they mean pick up. They will act on the pre-enteral follicles. The pre-enteral, we mean the late pre-enteral, يعني the late secondary follicles, to stimulate their growth into enteral follicles, into early enteral and then to late enteral follicles. Okay, so how do they, FSH and LH, what do they do? How do they act on the theca cells and granulosa cells? In fact, once the LH will bind to the theca cells, it will induce the synthesis of androgen by the theca interna cells from cholesterol and acetate. And in fact, the theca interna cells, under the action of LH, will produce androgens, testosterone and androstenedione from the cholesterol and from the acetate. And then the granulosa cells, under the action of the FSH, when the FSH will bind to the granulosa cells, will bind its receptors on the granulosa cells, this binding of the FSH to the granulosa cells will help in the transformation or the transformation of the androgens into steroids. Okay, they will activate, in fact, this binding of FSH to its receptors on the granulosa cells will activate the aromatase, the enzymes that will convert the androgens into estrogens. Okay, so the FSH binding to the granulosa cells will help in the synthesis and release of steroids, which are estrogens, okay, by the granulosa cells. So the FSH binding to the granulosa cells will help in the synthesis of estrogen by those granulosa cells. And this production will increase as the follicles grows. Now, the more we will have in fact, the more the follicle will the more this follicle is big, it will respond more to the action of the FSH, and it will produce more estrogen. Okay, so this production of estrogens is increased as the follicles grow, because when they grow, they will have more granulosa cells. So more granulosa cells, this means more receptors to the FSH. So they can respond more to the FSH and produce more of the estrogen. And then they will produce more of steroids, more of estrogens. We will have more production of estrogen in the biggest follicles, in the more mature follicles. Okay. Now the main estrogen produced are the estradiol 17 beta and the estrone, but also those granulosa cells under the action of FSH, they can produce the insulin-like growth factor 1 and 2. Okay, and those IGF-1 and 2, they act in a paracrine way on the theca cells, and they will induce the synthesis of androgen by those theca cells. So the IGF-1 and 2 produced by the granulosa cells under the action of FSH can act on the theca cells, okay? And on the theca cells, those IGF-1 and 2, they stimulate also the production of androgens by the under the action of LH, and they stimulate the androgen transformation to estrogens by the granulosa cells. So those IGF-1 and 2 activates also the aromatase enzymes, and that's how also they can stimulate the conversion of those androgens produced by the theca into estrogens by the granulosa cells. Now, here we can see a figure where we have the theca cells, and those are the granulosa cells. Now, the theca cells, they respond to the action of the LH, and they will produce, under the action of LH, they will convert the acetate and the cholesterol into testosterone and androstenedione. Some of those androgens will go in the blood circulation, but the biggest part will diffuse into the granulosa cells, where and those granulosa cells under the action of FSH can convert those androgens into estrogens, estrone and estradiol 17 beta. Okay, and those estrogens can be released in the blood. Okay, but also they can act locally in the ovaries, and also they will be released in the blood circulation. Now, the synthesized steroids are released in the blood circulation, and that, so to exert their endocrine action, since they will pass in the blood circulation. They have but also they have an important local intrafollicular roles. So not all of the steroids or not all of the synthesized steroids will pass in the blood to have an endocrine actions. A lot of the steroids produced will remain in the ovaries and will have a local intrafollicular roles. Okay, so they will have paracrine actions. In fact, those steroids, we mean the estrogens and progesterone. Okay, now here we're talking about the estrogens. They act with the FSH to stimulate the proliferation of the granulosa cells. So as we have said, the proliferation of the granulosa cells, we said that the growth of those of the enteral and enteral follicles is made under the action of FSH. Okay, so how the FSH induce the growth of the follicles? It will induce the division of the granulosa cells. Okay, the FSH will stimulate the mitotic division of the granulosa cells. Okay, and that's why we will have an increase in the follicular development under the action of FSH. Now also the estrogens, all the estrogens produced by the granulosa cells will act in a notocrine and paracrine way to induce also the proliferation of the granulosa cells. Okay, so the estrogens, the steroids produced by the granulosa cells will act with the FSH to stimulate the proliferation of the granulosa cells, and that's how they will stimulate the follicular growth. The androgens produced by the theca serve as a substrate for the conversion to estrogens, as we have precise, since the androgens are transformed or converted into estrogens by the aromatase. So they are the substrates and the estrogens are the products, okay? And those steroids will stimulate the aromatase activity, thereby promoting the estrogen synthesis. So the steroids produced also by the ovaries, they will stimulate the aromatase activity to help in this conversion of the androgen into estrogen and thereby promoting the estrogen synthesis. Now we have other factors produced by the granulosa cells, and they act also on the enteral follicle, and those factors are cytokines like the activin and inhibin. So the granulosa cells, under the action of FSH, they can produce activin and inhibin, and so their production are stimulated by gonadotropins during the enteral phase, and they mediate and modulate the actions of steroids and gonadotropins. We will see how the inhibin will affect the production of FSH and also the production of steroids by the gonadotropins. And in fact, those cytokines, they support the follicular progression, like the inhibin. Okay, this the inhibin, for example, it acts most in cooperation with the FSH and estrogens and androgens to support the follicular growth. Okay, and we have other cytokines like the anti-Mullerian hormone, the AMH, or the Mullerian inhibitory hormone. Okay, we have seen it in the chapter 3, the gonadogenesis. This cytokine is also produced by the granulosa cells, but it will promote atresia. It promotes the cell death of the granulosa, of the follicles, okay, of the growing follicles. Okay, so just you should know that those granulosa cells produce also other, they produce, they don't only produce estrogens and progesterones, but also they produce cytokines like the activin and inhibin and the MIH, and those cytokines play a role also in the regulation of this follicular growth, okay, and this of the steroidogenesis of the follicles, okay. So this is all regarding this session. We will continue next session. Do you have any questions?
No,
Are you
doctor.
still? Is it clear for you?
Yes.
Yes, clear.
Okay. Okay, we will continue next week.